Shall we? Sure. All right. Good afternoon, everyone, and thank you for coming. I have great pleasure in welcoming our MTS speaker today. It's Dr. Martin Weiner from George Mason University. But while we welcome Martin, I'd also like to welcome our guests from UC Davis, who've driven all the way from Davis, Will Joyner, and Weiwei, who've come down from Davis to listen to Martin talk. That's the power of Martin's appeal. Martin is a professor of psychology at George Mason University, and before this, he was a AAAS fellow, working on science policy, both at the National Science Foundation. And I've run into Martin over the years at many, many places, including small conferences, the White House, places like that. You know, as you may realize, Martin kind of gets around quite a bit. Martin got his PhD in 2011 at the University of Pennsylvania. He's been working on the area of timing and time perception. And he's, I would say, one of the rising stars in that field. Martin is one of the directors of the Timing Research Forum, an international timing society that meets biannually. Most recently, I saw Martin in Mexico about a month ago when I was at the TRF meeting over there. And today we're delighted that he can be here to tell us about his new cutting-edge research on dissociable and overlapping mechanisms for time and space perception. So please give a warm UC Merced welcome to Martin Weiner.
Thanks, Ramesh. That was great. I really appreciate that. I guess I could save it for the end, but I will kind of make the plug that there is this forum that I'm on the executive steering committee for, the Timing Research Forum. So, just to note, if anybody is interested in this kind of work afterwards, we do have a large list of people, students from all over the world, who are interested in understanding timing. Really construed broadly, anything from rhythm to language to music to tapping, things like that. And so if this is something you're interested in, I'm happy to talk more about that. Anybody is free to join. And every other year we do hold a conference. Our next one is going to be in 2 years from now, held in the Netherlands. So if anyone's interested, please let me know. But I'm going to talk to you about some of my work here today. The exciting thing about this, and the reason why I'm looking forward to telling you about this, is that this is all new, and none of it is published. So it's kind of in that raw, unrefined state where I'm really looking for feedback on things that may be missing. And I will also start by saying it's not perfect. There are certain little flies in the ointment in these data, and so that's why I'm particularly hoping to get some good feedback from the students and from all of you on this. But I'm going to start with kind of opening and saying why I'm interested in this area when I talk about time and space. It's that and part that's important. Whatever, I'll just push the button. There we go, okay. Okay, so I'm going to kind of set the stage opening with a paper that came out back in 1981 in Science. So this is one of these papers that kind of fell by the wayside. So it came out, it got some attention, and then it was gone. Like largely just remained a footnote. So this came out back in 1981. A guy named Alton DeLong, who was an architect in Tennessee, did this study where he brought people in and he had them play with small scale model environments of different spaces. And it might be like a little train station, or it might be a little dollhouse room, or it could be like a teeny tiny closet-sized room, all different sizes across all different people. And he just said, play with this room, imagine you're in it, and tell me when 30 minutes is up. Bye. And then he left. And what he found was that, by and large, people's experience of 30 minutes became warped to the size of the scale model environment that they were in, such that the smaller the environment, the shorter amount of time had passed before they said 30 minutes had passed. With the shortest amount being 5 minutes, a person was like 30 minutes was up and they'd been playing in this tiny little closet. And even though it varied from person to person, it still was consistent enough that he came up with this mathematical law that he called experiential relativity, suggesting that our sense of time is warped to the space in which we inhabit. And this finding actually did get replicated a few years later, and then after that just kind of disappeared. No one really kind of came back to it. But I'm using this to catapult into the world of space-time to say that our experience of the world is tied both to the dimensions of time and space. And in order for us to understand how we inhabit it, how we exist within it, it is necessary for us to understand both of these dimensions. These are, if you go back to the classics, Immanuel Kant in his Critique of Pure Reason said that time and space are the dimensions by which we interact in the world, and so in order to exist, we have to understand them better. So, time and space are big, so what I'm going to start out doing is kind of filling out our address here for what I mean when I talk about time and space. Certainly if we're talking about space, there's a whole variety of scales that you can refer to here. Everything from kind of small spaces to a room, up to very, very large scale spaces like a continent that you inhabit. And there has been some work that's just come out recently suggesting that the brain does represent in a gradient-like fashion, going from sort of very, very small to large kind of spaces across different gradients within the brain. Most of them are called the hippocampus and also in the parietal lobe. So there is this representation of space. This is distinct, however, from our representations of time, where we can have references anything from, say, a few seconds to milliseconds, all the way up to days and months, and then eventually years. And again, there has been some work on this that's come out somewhat recently, suggesting that when people are thinking about time on very, very long scales, and this is a study that contrasted two different types of questions to a subject, things like imagine yourself in Paris 1 year ago versus imagine yourself in Africa 5 years from now, that they were able to dissociate lengths of space and lengths of time to different particular networks in the brain. Most certainly for space, these kind of like more posterior regions, and time, these more anterior regions. But I'm talking about very long scales here. The area that I'm interested in is this short time frame. So the more immediate specious present. So here we're talking about seconds to minutes and distances from feet to meters. And the question becomes, why am I interested in this? Why do I think this is particularly important? And my argument is that this is the frame in which survival is most necessary. So this was from a paper that came out in Trends in Cognitive Science recently, and it kind of presented this idea of imagine you're a person and you have a tree behind you and you have a predator in front of you. You have to make a decision of whether you're going to run forward or over here or go ahead and climb up the tree. And the case is that the relevance of those actions are going to depend largely in the position in space that you inhabit relative to the crocodile and relative to the tree, and that both of these things are going to form a kind of peripersonal field around you for the relevance of those actions. And my case is, and they make this case as well in this paper, that time is part of the computation here. And then you have to say, well, how much time do I have to run away versus how long it will take me to get up the tree versus how long will it take the crocodile to get to me. And using space alone is not enough to survive in this situation. Okay, so it's kind of the last part of the history of this. This idea has been thought about before, so Vincent Walsh in 2003 looked at a number of fMRI neurophysiology studies and came to the conclusion that, well, at some point, the brain no longer cares about individual dimensions like time and space. It just jumbles all those things together and just calls them magnitude. It just says, well, I know this is small and that's big, and that's really all I care about, whether something is low or high or small or big. And it doesn't matter if it's time or distance or any other kind of discrete quantity. And he further made the case that this would rely upon a right lateralized network within the right inferior parietal cortex and the right dorsolateral prefrontal cortex, and these should mediate both of them. And this was back in 2003, and it's been fairly influential since then. But okay, if we want to really understand these quantities, let's just go to the map. So back in 2010, I published a meta-analysis of fMRI studies of timing. And so this came from tasks doing a variety of different methods for measuring time. So you can present subjects with 2 stimuli for different durations and say, well, which one of them is longer. You can have a subject mentally time something for you, so tell me when 5 seconds is up. You can also have a subject say, tap along to a beat or tap along to a remembered beat. And all of these studies kind of fed into this meta-analysis. And we came up with a number of regions. Most notably, the supplementary motor area seemed to be the most highly activated across all different study types. We also found a lot of basal ganglia and subcortical activation likelihood, in addition to kind of a more right lateralized network, such as the right inferior parietal lobe and right dorsolateral prefrontal cortex. And there was some success in this meta-analysis in mapping out the areas of the brain associated with time. So more recently, a paper came out in human brain mapping by Georgia Kona, who tried to do the same things but for space. And she included a number of studies in this meta-analysis, and these were things like mental rotation, spatial navigation, where you have to go from one position to another, and also spatial attention, so where you have to focus your attention from one position to another in space. And if you look at the areas that are activated here, and look at the areas associated with time, you'll notice that there are some in common between them. So the supplementary motor area, for example, does seem to show common activation between these two. We do get some subcortical activation, more so in the insula than in the basal ganglia, but it is there. Whereas we do see a bit of posterior activation for space, things like the precuneus, retrospinal cortex, hippocampus. So I reached out to Georgia Kona when this came out, and I said, this is really interesting. You know, we have this dissociation and overlap for time and space. What if we combine our meta-analyses together? So we started up a collaboration where we updated them for 2019, and we combined these 2 new meta-analyses together, and we said, okay, well, what are the areas of overlap? And it turns out that when you look at them, you find 3 regions that come out as being associated with time and space. We got, again, the supplementary motor area, the right inferior parietal cortex, and again, the right inferior frontal gyrus, roughly around the dorsolateral prefrontal cortex. So this kind of supports that notion of a common magnitude system in the brain that Walsh suggested. But we also asked, okay, can we take this meta-analysis and go a little bit further with it? So we have areas that we might say might be more activated for time, and we have other areas that might be more likely to be activated for space. And we can ask ourselves if there's some gradient between these two. So everybody loves gradients these days in fMRI imaging, and so we said, well, why not try and do something along those lines for ourselves? So when you take the map and you look at that, what you find is that in some of these areas there does appear to be these gradients between time and space, such that we see a smooth distinction from being more likely to be activated for time versus being more likely to be activated for space. And these do appear in, most notably, the supplementary motor area, where time seems to be more involved in a posterior sort of region, and space seems to be much more pre-SMA. We also see it, again, in this right inferior frontal gyrus to dorsolateral prefrontal cortex region for time to space, and again, in that right inferior parietal lobe. So we do see this substantial overlap, but it's not a perfect one-to-one mapping where you can say, well, the neurons in these areas code for both time and space, but instead seem to progress in some way between these 2 dimensions.
Excuse me.
Oh, yeah.
What's your task? I mean, how do you go from time to space?
There's no task here. So this is meta-analysis. So this is just taking studies that reported activation associated with space versus those that report activation associated with time. So there's no actual task. Well, there's a multitude of tasks that went into the meta-analyses. So this here is just saying, we just coded a study as being time or space.
So how did you define space? Is it big versus small?
No, so this comes from this work here from Georgia Kona, and so she put in a number of different space studies that she called coding for space, things like mental rotation, spatial navigation, spatial attention. The actual paper itself breaks those down into separate sub-regions. Same thing like my paper also breaks down the timing tasks into different sub-components. We were just interested in saying, is there overlap and is there a gradient between these 2 types of meta-analysis? you have a...
Yeah, I was just going to ask like what the data sources were. You kind of just said, like, so you just sort of curated different studies that you thought, you and her both curated different studies that you thought were either time or space.
Right, yeah.
How many about?
So in the time one, well, we had about, this original one had 41 studies in it, but I think as of 2019 it pushed it closer to 50. For space, there were many more studies. So I think this is on the order of like 100 to 110 studies at this point, just because more people study space than study time. But okay, so the question then arises, are these two things the same or are they distinct to some degree? So I'm just going to touch briefly, I know I said I'm going to talk about unpublished stuff, I'm just going to talk about, well, I'm going to publish data that just came out. This was a study that we did to try to differentiate between time and space in individuals. So it involves a spatial analog of a temporal delay discounting task. So this is a task where a subject is presented with, in a virtual reality environment with no landmarks, two kind of reward boxes, one that's close and one that's far, for two different values, one that's always going to remain fixed and low, another that's going to be variably high. And they have to pick one of those rewards and actually walk to go ahead and reach that. And we were able to vary the distance of that far reward across different distances. And we found predictably the farther that reward goes, the less likely a person is to pick it, unless its value is particularly high. But in order to further manipulate this, we did a second experiment where in addition to having this setup, we also changed the speed at which people could walk to get to that farther reward. And what we found when we manipulated the speed was that you could separately covary for either time or distance, such that even when you had rewards at multiple distances that could be reached in the same amount of time, people are more likely to pick a reward that is closer to them than one that is farther away. So even if something can be reached in 5 seconds, if something is closer, I'm more likely to pick it. And that suggests that there is this dissociation in that computation, because if it was only time, then it shouldn't matter how far away the other reward is, because if I can get there in the same amount of time, I'll still take it. And so this just came out in Cognition, if anyone wants to read that. But I'm going to go on to talk about the new stuff. So the new stuff comes from a task that was published in 2010 and then later in 2011 in Nature Neuroscience by Mehrdad Jadziri. And this is a very old task, but it was kind of presented in a new sort of way. And it's what's called basically a reproduction task. So what I'm showing you here is a spatial analog of the same task like for the delay discounting task. Except you're placed out in this open world and you're asked to walk for a certain distance, unknown to the subject. You're stopped at some position and you're then told, now walk the same distance again and mark it when you think you're there. So a person walks, you stop them, and they walk again. Crucially, what you do is you manipulate the speed at which they walk in both of these segments, such that a person can't just close their eyes and just like count or time how long they walk the first time and then do that again. There has to be some spatial component to it. And when a person does this task, what we find is a regression to the mean, where subjects are more likely to overestimate whatever the shortest distance was and underestimate whatever the longest distance was, and that scales to whatever the level was of distances presented. But this task has been done across a whole variety of domains. So this is here for distance estimation. It's also been done for angle estimation. It's also been done for time estimation. And here's one for length estimation. And across all these tasks, we see this very similar regression to the mean. And this has led people to suggest that these tasks, are all tapping into the same mechanism. But I'll note, however, that very few of these studies, if any of them, test multiple subjects on the same paradigm across different dimensions. So at the outset here, we want to know if we had subjects do this task and either had to measure time or space, would we see a dissociation either in the behavior or subsequent neural activation? So just to kind of present to you the way this task works, a subject's presented in this open field environment. They have to walk forward. While they're walking forward, that's the estimation phase. They are then stopped and are then told they have to reproduce the interval. And then that's the reproduction phase. And we did this for both space and time. So in the time reproduction version of this task, the subject is told, you're going to estimate time. They walk for a certain unknown amount of time, they're stopped, and then they have to reproduce it. And the durations here were between 1 and 5 seconds. Whereas for distance, it was the same thing, except they just had to walk, then they had to reproduce the distance, and they had to reproduce between 4 and 10 meters. And the reason why we chose these particular distances and times were they were basically normalized in terms of this is the distances that you would walk in these amounts of times, and this was the amount of time you would walk in these distances, such that people were experiencing the same distances and times in both versions of the task. And also just note, we did give feedback to subjects in the form of an adaptive window, so subjects saw a green sphere on the horizon when they reached that position if they were within a certain window. So here's just an example of what that task looks like. So I'm just going to shut it off. So see subjects kind of walk. So this is an example of a time trial. They're then stopped, and they're told they have to reproduce time. It's a fairly long attention interval. They walk again, and then they mark it when they get there, and then they get their feedback. In this case, it was good. For a distance trial, it's something similar. And what you'll notice too is that the walking speed is varying from trial to trial and segment to segment. So again, estimation and reproduction, the speed is always varying in some amount. Okay. So here's again the similarities and differences between these two tasks that I want to emphasize. So let's imagine you're doing this distance task. At the outset, you have to kind of then wait and then walk forward for some unknown amount of distance. While you're doing this, presumably you are referencing some internal odometer that's telling you how far you've traveled. Then at the start of the reproduction phase, you have to then go and decide, okay, how far am I going to walk? You then have to walk that distance and then reach your goal and say, okay, I'm there. In the time version of this task, it's fairly similar. You have to start out by kind of walking, except here you're referencing an internal timer or pacemaker in order to measure, okay, how much time have I been walking for? And then now at the outset, you have to walk for the certain amount of time, but because there's that speed change there, the distances are all scrambled depending on the amount of time that you're walking for. So you could walk for any particular distance as long as you're matching the right amount of time. So the first study that we did to kind of investigate these mechanisms was an Eeg study. So we had 16 subjects do this, and this was 64-channel EEG that we recorded. And there were a number of components that we were interested in here to kind of see if there were any differences in time and space. So from a frontocentral cluster of electrodes, we were particularly interested in the broadband ERP space with the contingent negative variation. So this is a negative signal that has been associated with time. It's also been source localized in the SMA quite frequently. On the frequency end, we were also interested in beta oscillations, so anywhere from 13 to 30 hertz. These have also been seen as kind of a frequency representation of the CNV. Whereas for space, we were interested in a posterior cluster of electrodes where people have shown before differences in evoked theta and alpha power that appear to be linked with navigation to some degree, particularly when you are doing path integration, as a person is in this case, that is dead reckoning. Like, I don't have any landmarks to guide me, so I just need to go until my sense of distance tells me I'm there. And there's some evidence that these two things are distinct. So this is, again, some work from Arne Ekstrom's lab, suggesting that there's a difference between kind of like frontal-mediated theta oscillations versus more kind of posterior alpha to theta power while subjects are walking around in a VR space. So okay, let's talk about the results for a second. So again, we've got time reproduction and space reproduction, and for both of them we see that central tendency effect. So if a subject was perfect, they would be responding along this dashed line, whereas instead what we find is that, and you can see it very clearly here for space, subjects are likely to overestimate the shortest distance and underestimate the longest one. For time, it's a little bit offset. We see much more overestimation for short intervals, but we do get an underestimation for the longest one. Crucially, if we take the slope of both of these lines here across subjects, we find that they're not correlated between the two and there's no difference So subjects don't show a higher degree of regression to the mean for space than for time or vice versa. The one salient difference that we did find, however, is that subjects are more precise in their estimates of space than they are of time. And this was surprising to us. Most subjects reported that the tasks were fairly equal. We didn't see this before in any pilot data, but we did find this difference in precision. I should note that being more precise doesn't mean you're more accurate. It just means you're more consistent in the responses that you're making. So, okay, but then the big question becomes, well, what do we see in the brain? So, for the CNV effect that I was talking about, that contingent negative variation, if we time lock to when subjects begin to reproduce the movement, so this is during that reproduction phase, what we find is that there is that CNV signal that has been associated with time that's significantly greater for time trials than for space trials. And you can see here in the topography like a much more frontal mediated effect going on there. So we do see this difference there in that CNV that is putatively driven by the SMA. But we can go ahead and break this down a little bit further. That is, we can say, okay, well, I understand that there's this difference in the CNV, but is there an effect that changes depending on what the distance was versus what the duration was that a subject was reproducing? And when we do that, what we find here is that that CNV effect that's occurring there in the beginning is modulated by the amount of time that the subject is going to reproduce. Whereas for distance, there doesn't appear to be any modulation depending on what a subject will go on to do. And there's a crucial point to this, which is, this is occurring roughly about 500 milliseconds into the trial. And that is well before any of the durations that the subject actually reproduces. And so before a subject has even reached their destination, there's a divergence in that CNV signal for what they are going to do. And I'll talk on this a little bit later, but this is consistent with some models of timing suggesting that when you're going to time something and you know what it is you're going to do, it's not that you just count your clock until you reach there, it's that you wind your clock faster or slower depending on the interval you need to reproduce. That is, my clock can only hit a fixed threshold. I'm going to make it run faster if I have to time something that's quick, versus now I'm going to make it tick slower if I have to time something that's long. And this fits with that pre-planning idea. Okay, but the next question is, okay, well, what is there in terms of a difference between time and space? So we can then look in that frequency space that I mentioned between those clusters. And here what we find is that when we look at the contrast between space and time, so in this case, anything blue is greater for space and anything red is greater for time. If we look at that frontocentral cluster, what we find is there's this increase in this broadband effect, roughly spanning from alpha to kind of like high beta, but it peaks right around kind of like 18 to 20 hertz for a beta oscillatory effect that's happening specific for time. Whereas for space, what we find is a later cluster that's coming from posterior electrodes that spans, again, that alpha to theta range that Arne Ekström has been talking about as being associated with spatial navigation. And both of these are coming from that reproduction task where a subject is actually going to initiate that movement. So it does seem that we have this kind of pivoting difference between these two tasks and these two frequency ranges. And it's kind of like a final thing that we looked at. We did check out frontal mediating theta. And here we were looking at, is there a difference in intertrial synchronization, or called intertrial coherence in the EEG lab terminology. And there what we found was when subjects are, again, initiating this task, there is this increase in intertrial coherence for theta oscillations associated with time but not with space. Oh, and I should also note this was only observed for when they're beginning the trial, the estimation onset. So, okay, so overall what we're finding is that subjects are more precise at reproducing distances at time, but that doesn't mean they're more accurate. We see that timing mediates this larger frontal CNV signal associated with beta that also seems to be split depending on the duration of your timing. That supports that pre-planning hypothesis. And the spatial reproduction evokes a larger posterior theta to alpha power during consolidation. Oh, and there's this FM theta effect. Okay. So we said, all right, we see this difference in the EEG, but what we really want to now tease apart is there a difference going on at the level of Fmri? That is, we have the meta-analysis results. Can we go and confirm this? Because again, the meta-analysis came from different studies, studying different hypotheses, studying only space or only time. Well, what if we did a study that looked at both together? So we took 24 subjects and we had them do the exact same tasks that I told you about before, the time reproduction task and the space reproduction task. We had certain areas of interest, so for example, it's been recently shown in the SMA with 7 Tesla MRI that there is in fact a gradient of activation in the SMA depending on the length of the interval that a subject is supposed to be perceiving or detecting, where we see more posterior activation for longer intervals and anterior activation for shorter intervals. Likewise, around the same time, there's been work shown in the hippocampus suggesting that there's a difference in spatial processing fields for very, very long distances to very, very short differences across the hippocampal long axis. So I should note, however, that I did have subjects do the spatial version of this task previously. So this is back in 2016. This was only the spatial version, just spatial reproduction. And there we found a fairly widespread frontal-parietal network that was associated with spatial reproduction. However, we're coming back to this now because we don't know how much of this is associated with time. That is, subjects could still be perceiving time while they're doing this task. Again, remember, I'm saying that time and space are intertwined and that you have to do specific manipulations to tease them apart. So first I'm just going to touch on the behavior. Again, we see something very similar where there's this central tendency effect. It looks a little bit different from before, where now we see kind of more underestimation at the longest intervals, and we don't see a very clear difference between the sort of tasks in terms of that level of central tendency. Also noteworthy is there is no difference in precision here, and I have some speculation on this as to why, if anyone is interested afterwards, such that subjects are not any more precise for space than they are for time. But all right, let's go to the map and talk about the activation. So for time, we find kind of this frontoparietal network that includes regions like the supplementary motor area, the right inferior frontal gyrus, the right inferior parietal cortex, bilateral representation of some of those structures. If we go and we look at space, again, we find something very similar, including the SMA and the right inferior frontal gyrus. There's a bit more posterior activation, but we see both of them. So this is consistent with what we had seen before, and it is to some degree consistent with the meta-analysis. But the more interesting thing is if we contrast between the two. So this is again for time versus space. And here when we do this, we find something particularly striking. In that, if we're contrasting between time and space, areas more active for time and more active for space on this task, the only area that comes out as being active for time is the supplementary motor area, represented bilaterally. That is, it is the only region that comes out as significant. Whereas for space, what we find is this clear demarcation of anterior to posterior, where we see this posterior network of striate and extrastriate regions across the occipital cortex up to the angular gyrus and parietal cortex. There's also, you can't see it too well here, hippocampal activation as well. So we see this very, very clear distinction between time and space when we contrast the two of them together. Notably, with time only bringing out one area and space bringing out this more, again, posterior network. Now, if I were to kind of wildly speculate here, I might notice that there's this distinction between, say, the SMA and the precuneus across kind of like superficial parts of the cortex. And I might notice that there was this dissociation in the EEG between beta oscillations that were happening early in the anterior cluster and theta to alpha oscillations that were happening later in the posterior cluster. And I may say that those two things are part of the same representation. But I didn't actually do any work to kind of look at this, so I'm not going to say those things. Okay, so we've identified where some of these regions are and to a degree when they're involved. But this is different than actually saying what it is that they're doing. We can identify and we can map brain regions, but it's another thing to go and say, well, what are they actually doing? What are the computations they're involved in? So for time, when I talked about that 7 Tesla MRI study where you have different parts of the brain associated with different intervals, this fits with a population coding model of time that has been proposed in the same way that the brain uses population coding for other dimensions like angle and to a degree numerosity, where you have to have neurons that are tuned for specific durations. That is, you have to have, say, a 1-second neuron that's right next to a 1.1 second neuron that's right next to a 2-second neuron, and so on and so forth, both upwards and downwards. So that's one way in which people have gone ahead to explain this with the population coding model. So to try and tease this apart, we ran a decoding analysis using a 12mm searchlight, where we fed into this thing all the possible durations that subjects were presented with, and all the possible distances that they had to reproduce. And we sought to find out if we could decode what either the time was the subject was experiencing or reproducing, or what the distance was they were experiencing or reproducing. So, as a first demonstration, what I'm going to show you here is what happened when a subject was estimating the interval. And for the estimation phase, in this case, we are time locking these effects to the offset. That is, when you are at the start of an estimation trial, you don't know how long the interval is that you're going to be walking for. You're just told to walk until you're stopped. So what we've time locked to here is the moment when everybody got stopped. And so here we're going to see, okay, you only know it's 1 second when you reached it, when it's elapsed. And we sought to find out, is there anything there that we can use to predict what the actual interval was? And so over time, if we look at time decoding versus chance, we find significant activation or significant decoding in a number of regions, including the supplementary motor area, also again bilateral, inferior parietal or superior parietal cortex, also left premotor and a little bit of ACC. And it's great to kind of demonstrate this decoding versus chance, but if we want to kind of like turn the dial up even more, we can say, is there anywhere where we can do time decoding better than space decoding? And when we do that, the only area that comes out being significant is the SMA. That is, it's the only region where we can decode the interval that the subject has just estimated, consistent with that population coding model presented before. And if we look under the hood of that SMA cluster and kind of like plot the confusion matrix, we can kind of see how well it does. And if this thing were perfect, it should like perfectly classify along the diagonal here, the true duration and predicted duration should match. And we find that it does, fairly well for some of them, particularly the longest duration, but along the line it gets kind of confused. In particular, for the shortest duration, it makes a lot of mistakes and overestimates them. Whereas to some degree, for some of the others, it underestimates them. This is interesting because it seems consistent with that central tendency effect that I was telling you about before in the reproduction data. So okay, what someone could say here, and this has been said before, is, well, could you just be measuring that accumulator? Could you just be measuring that pacemaker that I was telling you about before? And if that were true, then the activation should just get up or go up for the longer intervals. That is, the longer the interval, the greater the activation. And so it should go in this kind of smooth, progressing gradient. Well, if we take a look at the beta values in this cluster across the different intervals, what we find is there is no difference across them. So it doesn't seem consistent with an accumulator model. It instead fits again with this pattern of activation that's being decoded at the population level, akin to population coding. Now, all right, I've told you guys about that pre-planning hypothesis, so what if we take a look at what happens when subjects are reproducing the interval? And here, I'm time locking to the start of that reproduction phase. That is, when a subject is about to start reproducing. And the question is there anywhere where we can decode how long the interval is the subject is about to reproduce? And when we do that, the only area that comes out as being significant is, again, the SMA. And here, when we go ahead and take a look at the confusion matrix, we actually find much better performance now for reproducing intervals, where we get short and long intervals that are encoded very well, but some middle ground there for the middle intervals, the ones that should be the most uncertain or the most difficult to kind of classify for individuals. And sure enough, our decoder finds that difficult as well. But I mentioned to a degree some flies in the ointment, and so I'm going to mention one of them. So there was that estimating time case that I made to you here where I said, well, that's not an accumulator. But if we look at reproducing time, here we find that the activation does go up, where the longer the interval, the greater the activation that we see in the reproduction phase. But I will say not so fast, because this is actually exactly the same thing that we found in the EEG data. Where remember, this was at the very beginning of the reproduction phase before they've actually reproduced the interval. And so here again, we're finding the same kind of thing coming from both clusters, showing that there is this preparation to go ahead and reproduce an interval of a specific amount of time. And I'll note that there is some work also showing, so this is very recent work that just came out in Neuron by Meredith Jaziri's work, showing the exact same thing when you record from SMA neurons in primate cortex, and you make them do that exact same task. That is, if you look at the kind of latent cortical dynamics, and so here they just recorded from hundreds and hundreds of channels and just run a PCA analysis and projected it into kind of the first 3 PCA components, what they find is that there is this division across the basically cortical trajectories that divides itself up depending on the interval that the animal is going to reproduce. And again, they've modeled this now with a neural network model that matches this idea that you have a fixed threshold of activation that's being met, and really what you're changing is the rate of accumulation that's going to occur, this kind of pre-planning. So, okay. The overall findings of the Fmri is that subjects are less biased when reproducing distances of times, but not more precise. Time and space reproduction both evoke frontal-parietal subcortical networks. The time seems to specifically engage the SMA, whereas space specifically engages these posterior striate and extrastriate regions. And then lastly, we can decode intervals from SMA activity patterns. So, okay. I could end it there, but I'm not, because there are, again, some other things to talk about. Because probably, hopefully, everybody here is wondering something at this point. Because I've kind of gone through this and told you these things, and now I can wave and say bye-bye. But some of you are probably going to say, what about the hippocampus? You just made this great case saying that the SMA is going to code for time and the hippocampus is going to code for space. So why aren't you showing us anything decoding for space? And the answer was, I couldn't do it. So I tried looking in the hippocampus, and I tried seeing things, and I found just nothing. Nothing is coming out for space for decoding. And so this is just kind of demonstrate here accuracy versus chance for left and right hippocampus ROIs, and see is there anything there? And the answer is there's nothing. There's a little bit above the level of chance, but we're not finding anything that's consistent and nothing that I want to hang my hat on. But if I go ahead and say, all right, fine, maybe I'm setting my threshold a bit too high here. What if I just look at p less than 0.001 and I don't worry about cluster correcting, which some people do when they present these studies. They say, well, we had a 5 millimeter small volume correction in this particular area, and we found it. And I could do that here, and so that's why I'm just going to present it to all of you rather than make the claim that I have. So it turns out if we look at distance estimation onset, we can decode the distance that the subject has just perceived, but it's not involving the hippocampus. Instead here, the areas that we find that are most notably involved seem to be the anterior cingulate cortex and the right cerebellum, where we find 2 clusters that seem to be particularly relevant for decoding what the distance was that the subject had just experienced. But I'll note to you that, again, this is uncorrected. But okay, what about reproduction onset? Is there some similar pre-planning that's going on when you're preparing to walk a particular distance? Well, when we do that, again, we find 2 regions that are not the hippocampus. We find the retrosplenial cortex and the left caudate. And both of these regions have been seen as highly relevant for navigation. And if I'm going to note one thing here, it's going to be about this particular area here, the retrosplenial cortex, because this is an area that, just like the SMA, people in navigation have been asking for years, what does this thing do? In fact, there's a lot of debate about what the RSE could be doing. Just like in time, we're asking what the SMA could be doing. And I will note that in my 2016 study on distance, we also found the retrosplenial cortex as being significantly associated with behavior. And that was that degree of central tendency that I told you about, that regression to the mean, covaried between subjects with activation in the RSC. And if we look at where these regions where I'm finding decoding, and where I also found this association between subjects for central tendency before, they do appear to be occurring in relatively similar areas of the RSC, with the one note, if anyone catches it, that these are actually different hemispheres. However, just like the SMA, we find bilateral activation in the RSC. They're right next to each other, and so it's an open question whether or not these are reflecting the same process or some similar process. I've scoured the literature. If there's a navigation person here who can tell me there's a case for left versus right RSC, I would love to hear it. So okay, let's go full circle here and talk about what I've been saying, which is about decoding phenomenological space-time. That is, I can decode time very easily in the brain, but space is hard to decode. And that's not what you might think. Well, the question is, well, so why? And what I'll note is that when you're doing this space task, if you are involving, say, the retrospinal cortex, there's actually a variety of strategies that you can use to accomplish it. Most notably, when you're doing this task, you have all the environmental cues that are available to you. These include the optic flow of how fast you're traveling. They include information about the world in which you inhabit. They include an awareness that's immediately accessible to the individual of saying, that's over there and that's where I need to go. And that's different from time, whereas you don't necessarily project out there, okay, that's the time cue that's going to lead me to this thing. Time is rather an internally mediated process where subjects have to actively disengage from space in order to measure the time that they have to walk. And when that happens, I could argue that in fact it makes it easier to decode things for time because I'm just using this one process, whereas for space I'm using this variety of processes. So it seems like space occupies this kind of parietal or retrosplenial source that's associated with orienting yourself in the world. And that seems to be what the RSC is doing. That's the story that people have made about it. Whereas for time, there is this coincidence detection mechanism where I have to take the SMA and everything else that's going on and use it to track a particular metric of how long something has lasted. And these two things may represent very different mechanisms for measuring and understanding time and space in the world that we inhabit. But the next question becomes, where do these two things interact with one another? And that's the area that I haven't yet solved and haven't yet approached, but it's the next phase that these data are meant to lead to. So I'm going to end it there. I'm going to say thank you to Ramesh for inviting me out here. Thank you to everyone as well for listening. It's been great talking with you all. I just want to thank my collaborators, including Georgia Coda and Giovanni Mioni at the University of Padua, and also thank the National Science Foundation who funds my work, and my lab and everyone here. So thank you very much.
Time for questions, beginning with questions from grad students first.
For the decoding space, have you tried looking at hippocampus and plotting it from posterior to interior axis, and you still?
I have, yeah. So I tried also dividing it into distinct subfields from anterior to posterior, and there's nothing.
Okay.
Yeah, there's just nothing.
In the studies where this is reproduction task, this is all in virtual reality. Is the distance and time and all that set by the program, or is it something where the participants themselves are sort of moving, ambling along? Because I'm wondering how participants are at estimating time and distance when using sort of their own natural gait and their own sort of internal sense of time.
Ah, okay, like I see. So like if a subject were actually in the world.
Yeah, I don't know if I'm phrasing it correctly, but you seem to.
Yeah, so I would suspect that there is a difference between those two things. I mean, subjects are not receiving any vestibular input. There's nothing telling that they're moving. There's just that virtual appearance of movement. And so there is work. So our Ekstrom's work group is doing this. There's another group, I think at UCSD, that's doing this as well, using omnidirectional treadmills associated with virtual reality and EEG or mobile EEG. I'm not sure what those studies have found yet in this space. I think the work there is still pretty preliminary, but they are suggesting that they are different. Although at the same time, when subjects do virtual reality tasks, whether they're walking around, moving around, even with a joystick, there is this somewhat anecdotally reported feeling of embodiment in that virtual world. And so the idea is, and it seems to progress the longer you are in it, the more you feel you are inhabiting it. So I will certainly admit these are not perfect ways of measuring the actual brain responses for it, but they're what we have. So.
At the end, you were talking about this sort of idea of switching between the maybe default spatial reference stuff into some time-oriented thing, and I'm wondering about, in the spatial case, I think you mentioned that it also seems pretty obvious that there's a point of reference that you've got, and that you can constantly use the environment, this point as a way to help you do that calculation of what pace you're going at or what the rate is or whatever.
Right.
With the time, you have this weird thing that maybe is represented by that dip at the beginning where you just have to kind of decide what the pace is at the beginning. I mean, is there do you think that how much do you think that the fact that there's that the reference that you have for the time keeping up with is just all internal is relevant here, or is it all internal?
For time?
Yeah.
Time seems to a degree to be better if you're unencumbered by environmental stimuli. In fact, we found that your perception of time is largely tied to the stimuli that you are using in these different tasks. So there's a number of different tests that show that larger stimuli on a screen appear to last longer than smaller stimuli, that bigger numbers appear to last longer in time than smaller numbers, that higher pitched sounds seem to last longer than lower pitched sounds, or if there's at least some difference between them. Whereas if you're just timing nothing, if there's so-called empty interval timing, you may not be totally accurate, but you'll be far more precise. That is, to a degree, your perception will be more consistent if you're just using these internal mechanisms. So I would say it probably, to a degree, does rely on that internal timekeeping system. Although there have been some people who've made the case that in that case, what you're actually timing is your awareness of your body. Which, there they've speculated that you should see insula activation if that's the case. But we don't particularly see that here. But it may just not be the right task for it.
It's kind of random, but would it be fair to say that some of what you're trying to do is some form of intensity matching between these different dimensions? And if so, would any of the literature on intensity matching in other sensory modalities give you some insight as to how you could investigate what's going on in the brain for this?
Yeah, so when you say intensity, I think magnitude. Is that what you're...
Yeah, how loud is this sound relative to how bright this light is or something like that?
Oh, across modalities.
Yeah.
Yeah, people have done that for time, and largely they found that your sense of time does depend on the modality that you're using. So, for example, tones are perceived as longer than lights, but you only see that when you're comparing tones to lights, not when you're comparing tones to tones or lights to lights. You have to have these differences there where you start to see that there are these dissociations in time. But it's not clear, the debate in the literature is, well, does that mean I've got a little clock for lights and another clock for tones and another clock for my body and another clock for something else, and they're just running at different rates and I only see problems when I compare them? Or these things are feeding into a central mechanism that's just taking the signals in the best way that it can and trying to map time onto them. And it's not clear right now. It largely seems to be, both seem to be true to a degree, as with anything. Yeah. So. Yeah. Okay.
So, with all of the overlap that we see in timing and spatial processing, sometimes I'm wondering how much of it is that we're using time as a way to help us understand space, as in, especially when we're moving.
Sure.
Right, and vice versa. You can see how much we've moved, and that's a cue on how much time has passed. And so then I'm wondering how that would have an effect on the kind of task where you have where people are moving. So then my question is, would you think you would see a different kind of result if your subjects had to judge space without any movement?
Judge space without?
Judge distance without looking like how far it should be.
So they just like see a distance and they have to judge how far it is?
Yeah.
Yeah, we've thought about doing that experiment. The thing that that I suggest is that a subject has to have some familiarity with the environment in order for that to work. We did a study a number of years ago where we presented stimuli with an implied distance. And we found that it really just seemed to kind of subjects became very variable in how far they perceived the implied distance. And ultimately we decided it was because they were not familiar with the environment in which we were showing them these things. So what we want to do is a task where a subject experiences an environment. Say they walk around a whole world, and then we present them with snapshots of these distances or time, or I guess distances, and ask them things like, how far is it to reach this? Is this one farther, or is that one farther? Or would this one take more time, or that one take more time, in a kind of comparison task? But again, we think that it would be tied to actually experiencing that environment. And there's been some work to a degree on that from a group at Donder's, forget the name of the person, but there has been something close to that.
Couldn't you just use normal photos of the world instead of having it be a virtual environment?
Sure, yeah. And again, in the spatial navigation world, this is what people do. So like Russell Epstein's lab at Penn does this all the time, where they show Penn students pictures of things on campus, and they just ask them which one was farther. There's been a lot of groups like that. And that's where a lot of this work shows like the hippocampus being associated with distances, like parametrically associated with farther and farther distances. But, again, the debate then becomes, well, what is the hippocampus representing? Is it actually representing that distance, or is it representing the landmarks associated with that distance and the context in which the subject is inhabiting? I think to a degree that's what Iva Brunek's work was showing in the current biology paper that I referenced before, where she was showing people pictures of Toronto of like different distances and locations and stuff like that. And then she showed this gradual progression from short to long across the hippocampus long axis. But again, I think that the reason why she sees it and I don't may be related to the fact that this is an environment that subjects are very familiar with. There's lots of landmarks, and there's an easy way to situate yourself. Whereas I'm placing people in a wide open field of white noise where there's nothing to reference to, and then just taking them out of it and throwing them back into another random point on it. So in that case, you're completely dead reckoning. And there the hippocampus may not be as involved.
I'm just curious, I'll follow up on that. What do you think would happen if you took people, instead of taking a place that like Toronto has landmarks, and assuming she's using people from Toronto, if you took pictures of New York or some people who live in LA, right, who've never been there, don't know what those distances, don't have any memories of them, but they can still use their real world intuitions about how far things are. What do you?
Like a novel environment, you're saying?
Yeah, a novel environment with the same systems that you're used to.
Yeah, I don't know if anyone has done that. Someone might have. Certainly I know that they've done studies in recordings in rodents at the outset of being exposed to an environment they've never been in before, and then gradually learning that and seeing that certain regions are non-selective initially, and then over like a 24 to 36 hour period acquire selectivity for different distances and objects in that environment. If you took people from Toronto and showed them distances in New York, I could see either, it depends on how you would do the test. If you just said, how far away is this from you? A person might be able to say, well, that looks far away. Versus if you said to someone who's never been to New York, where's the Statue of Liberty relative to like the Metropolitan Opera? Like they might have no idea, and so they wouldn't be able to provide an accurate answer unless they tried to just conceive of a distance. But you probably wouldn't be able to decode it in that case.
Thank you.
I just want to make sure there was another hand. What about if you have settings where there's moving objects in your environment, right? Like where time and space interact a lot more. So I'm thinking of an example of driving down the freeway, and I'm thinking of a car in front of me, and I think 1/1000 distance, right? So my estimate of space is sort of an estimate of time. Like how far will it take for me to get to that object? Do you still think you'll see, in some way, I've kind of externalized time to a space metric.
Right.
Do you still think the SMA will be involved in that, or like since you've externalized it, maybe some other form of embodiment would take over?
I would think the SMA would be involved in something like that, because my argument is that in many cases when we're thinking about space, we're actually thinking about time. Like if I'm on the plane and I say, well, how much farther do we have to go? What I'm really thinking is how much longer do I have to sit here? You know, it's like, it could be the same thing as just saying like, okay, like, you know, how far until I hit that car? Well, really it's how long do I have? Like, and so the minute you extract from that the temporal information, that's when I would imagine the SMA would become most relevant. And to a degree, remember at the outset, I'm pointing out that, you know, the SMA is not only activated for time and not space, it's activated for both. Like we see it all the time in both sets of studies. My argument is that the SMA is incorporating both types of information into it in order to come up with this type of calculation that is time-based in some way. And I only find it here because I've, to a degree, dissociated those two things. So, yeah.
It's an excellent talk, and I enjoyed both Eeg and Fmri result. My only issue is with the experimental designs. So, Jordan had very excellent questions in terms of reference.
Mm-hmm.
So for your distant task, the reference was the distance. For your time task, your reference was distance, was not a time. So I think for me to be convinced that the results you're showing are related to time, I think I would like to see a task where both the distance and time are reference to the time. For example, a far sound versus close sound, estimated distance, for example. And if you show the same result, then I would be convinced that SMA is involved. My second question is, your result showed difference in difficulties, right? So the time was more difficult, which I'm not surprised. So SMA is related to error processing, you know that.
Mm-hmm.
So I think for me to be convinced again, both of them have to be equal in difficulty.
Right.
Or maybe you can reverse the difficulty and you see if SMA is activated for space task more in that case. Then if it's activated for more difficult space task, then that would show that SMA is not related to time.
Right.
So those are just suggestions. Since you asked for feedback.
No, sure, great. You found the flaws. That's great. Well, so I think that's great, and I agree with you. I'm going to push back on two things just slightly. So one, what I'll note is that so you said in the time task, what you're using a distance. So there is the fact that the speed is always changing. And so you can't use the distance when you're doing the time task because it won't provide you with any information that will help you in that case.
But the result you're showing is before you start moving.
It's 500 milliseconds after the person begins to move. So a person begins to move, and that's when we see this response happening. So like it's because it's time locked to the initiation of movement, and then we see it half a second afterwards is where we see that divergence in the activation. So I would argue that by having the speed manipulation, distance cannot help you in time, and time cannot help you in distance. And we actually also did an analysis where we looked are longer times associated with longer distances or longer distances associated with longer times. And we found that there was no connection between those two. If they were linked, so if I can only walk a longer time by going a farther distance and vice versa, then there's a strong correlation between the way that those things work, or if we parametrically modulate them. To your point about the difficulty of the space task, one other thing that I will note is it was only more difficult in the EEG version. In the Fmri version, the difficulty actually the behavior wound up being the same between the two. I kind of gloss over that. My speculation about that is that in the EEG experiment, time and space were blocked. So they did a block entirely of space trials, then a block entirely of time trials, whereas in the Fmri version, it was event related. So any given trial could be a time or space, and they were pivoting back and forth. And it seems that when you're just like, now it's time, now it's distance, distance, now time, the difficulty difference between them went away. Whereas if it was only a block of time versus only a block of distance, the difficulty disparities seem to come out more there. So I'll note there is that difference in the EEG data, kind of between the two of them, in terms of the difficulty. But in the Fmri one, not so much. But I do agree that certainly there is more to do to dissociate these two things. Another thing that we're thinking about is taking that delay discounting task that I mentioned before, and seeing if we can tie that to activation differences, because there we can pretty clearly dissociate between time and distance in terms of reward processing. So. Yeah.
On that question though, in the time task, you might not be able to use distance to help you, but aren't you still using distance to notice that you're moving faster and to know that you have to estimate time differently?
Well, again, it doesn't matter how fast you're moving. All you're trying to say is how long have I been moving, whether I've been moving a short of, fast or slow. So again, the distance doesn't really have a factor that influences you there. There are optic flow effects, but again, we tried to kind of like look into that. We didn't see that there was faster speeds were associated with longer intervals, as one might expect. But I think it was strictly because there was that decoupling of strategy, that a subject knew, I can't use time here. And so we told subjects ahead of time, we said, okay, when you're doing the time task, don't count it in your head, don't rely on any kind of metric to do this, and you have to do this in timing tasks. You have to say, just go with your gut instinct for the time. So to a degree, it seems like the speed should not have had much of an influence, or the distance.
Was just trying to remember back from earlier in the talk, where I got the impression that the EEG tasks, it was like a VR setup, or is that not correct?
The delay discounting one was VR. Okay. In this case, it was just a really very large monitor that was very close to them.
So then, the Fmri version of it, that's the same thing, except they were laying
flat, and they had a little reflective mirror screen.
So as someone that's, I don't know much about spatial processing, but does that orientation difference, would that have an effect?
I'm sure it does. Notably, when we had subjects do the Fmri version, we kind of debriefed all of the subject. Some subjects said oh, I got really disoriented because I was lying flat, but I saw myself walking forward, and so when you took me out, I was all turned around. Whereas others said, oh no, I knew I was I didn't feel like I was really inhabiting this world. I felt like I was on my back. So I think there's individual differences there from one subject to another.
Yeah. So related to that point, did you look at the data in terms of subjects that felt like they were more immersed?
No, we didn't. Yeah, you're saying see if the people who felt embodied in the world behaved or looked differently than people who did not. We haven't, but I mean, we could. That's a good idea. So.
Yeah, and I'd also be curious, I know you mentioned this at the end, just in terms of how the time and space networks kind of interact with each other, because obviously space and time are ingrained in our everyday life. Even some of the tasks that you mentioned in your meta-analysis, for example, the mental rotation, that's
space changing over time. Basically.
Right, yeah, sure.
And so I'd be really curious also to see if, to learn or hear about individual differences in maybe connectivity between the SMA and these more like precuneus or posterior.
Yeah, absolutely. Yeah, that is kind of one of the sort of next steps for these studies, these analyses is, well, how do these things interact? Can we do connectivity analyses between the two of them? And can we tie that to individual differences in how much a subject might feel embodied within that particular space? Those are open questions, and I'm like excited to look into them. But yeah, so yeah.
I think I understand why you're varying the speed, as you said, but do you have actual data about what the imaging data look like if people can predict the speed? That is to say, can you contrast physiologically what's happening when they're able to predict the speed and therefore could have this kind of confound between space and time?
We don't, but we could do something close. So the way in which the speed was manipulated was, at the start, we drew a possible speed from a uniform distribution. Then we altered it by a certain amount drawn from a Gaussian distribution.
Zero mean?
What?
Zero mean?
Yeah, zero. Well, no, it was actually kind of a truncated Gaussian, so it couldn't be zero. It had to be a difference, up to positive or or negative 60%. So max 60% faster or slower. But we can look at trials in which it was very similar. So in which the speeds were very close versus ones in which the speeds were very different. And we could see if on a trial by trial basis, ones where, because subjects knew that there was this speed manipulation. But we could still see if there's an effect for when you're walking and, oh, the speed is exactly the same. I'm just going to use time now because that's the easier strategy for me to do this. So we could do that analysis. It is possible. And I think that could be something really cool to look into. But you're saying then, I mean, the whole reason we did that manipulation was so that people wouldn't use, one strategy for the other. But I guess so. Okay, so yeah, so we could use that to see if there is this dissociation between the involvement of these anterior to posterior regions, right?
Yeah, and I think you're right. The suggestion of just looking at trials where the speed doesn't change much would be interesting. But I think it also would be interesting to, essentially break the paradigm and have them know what the speed is at the time that they're encoding.
Right.
Just to see what's going on.
Sure. Yeah. Yeah, we could do a separate manipulation like that where it is constant between the two of them.
So in the odometry literature, there's like a very strong specificity of encoding that happens in your report apparatus, right? Like, so if you, in tasks like what Millie was talking about, where you actually have to really navigate through a setting instead of in a virtual environment, your gait of how you walk to a target determines how you've encoded that distance. Because there's probably some things you've done in the way you kind of move yourself to that target that have given you this sort of embodied representation of how far you've gone. Right. So I'm just wondering to what extent, and in that representation, space and time are like completely intertwined. So I'm just wondering, in a context where you cannot, in an experimental way, kind of dissociate space and time, where they're totally embedded, isn't it kind of natural that the neural representations for those things probably kind of co-evolved?
Oh yeah, yes, absolutely. And so that was why I referenced that Iannetti Tix paper before, talking about the predator and like, well, what am I going to do? Am I going to like run away? Am I going to run up the tree? And their case was that warps the peripersonal space around you based on the value of a particular action. So it's going to say the value of running up the tree is much greater if the predator is very close to me, versus the value of running away is much greater if the predator is farther away from me. So to the degree that these things likely did co-evolve, I would say, it's probably assured, because it's necessary for survival in that case. Again, this is that idea, there was this idea proposed about the SMA a number of years ago that it serves as a vector transformation, which is the same, that is linking things like mental rotation to reaching to anything manipulating objects in the environment. in the sense that when I have to, you know, do anything, perform any action, I have to calculate a number of different sort of qualities about that action. Like, okay, in order for me to reach the cup, I have to precisely know how far I have to reach. I have to know get the velocity of my arm just right so that I'm not moving too fast or too slow. But I also need to like calculate how long it will take me to accomplish that action. And those three things could be separate computations happening kind of in different parts of the brain, but they need to be brought into a common space to actually enact or actually perform the action. And it was proposed that the SMA is that common space where these things are brought together. And so that, again, that might still be true. I don't think my data speak against that. So I think they likely did co-evolve, but they required coordination.
So where are some of the other regions where these things, so what about parietal areas, which also seem to be involved in these sort of sensory motor transformations, right?
Yeah, so there seems to be this dissociation between left and right parietal cortex. You know, I mean, you know, people talk about the left parietal lobe being involved in so-called motor attention, so attending to actions versus, you know, in the right parietal lobe attending to space.
Right, because when you get damage to the left parietal, it leads to sort of things like apraxia, and right leads much more to
spatial
neglect and
neglect.
things
Right,
like that.
yeah, exactly. Although for neglect, you see deficits in time as well.
Right.
But with apraxia, I don't know if there's been any work in apraxia and timing deficits. In fact, I don't think anybody has done that. Actually, I don't think anybody has. Not that I'm aware of. So that might be something to look into. I think there has been work in, well, actually no, there was a study that looked at left parietal lesions, people who had left parietal lesions without apraxia, like specifically without apraxia, doing a temporal attention task. But this was just like a 4-period task where press a button as soon as the target appears, and it could appear either early or late, you don't know which. And the idea is that if it hasn't appeared early, it must appear late. So you're faster when it appears late. And people with this left parietal damage were just impaired at that. They were kind of equal at both, if I recall correctly. So, yeah. So there are these other regions that are involved in the computations, certainly as it relates to kind of bringing them into some kind of, might be bringing into a common space like I mentioned. I should note that SMA lesions are themselves particularly rare and not often seen. There's only been, I think, like a handful of papers that found SMA lesions and tried to test people on tasks.
And it leads to a wide variety of crazy, unpredictable consequences, right? And even stimulation of SMA in the primate brain by Graziano and others.
Oh yeah.
Leads to very strange behaviors, nothing sort of predictable.
Right.
So there might be something very interesting sitting there.
Right. Yeah. Exactly. Again, so my area is asking what does the SMA do? Just like in spatial navigation, they ask what does the RSC do? So we each have our 3-letter structures that we're trying to disentangle. So. Yeah.
Perhaps I'm being naive, but I can imagine animals doing some tasks that are very close to this. And so if I had an animal do that, but I completely removed the hippocampus, is your prediction then that it wouldn't see distance deficits? Or?
I mean, I think we know that's not the case.
Right.
You know, like they do.
You weren't seeing activity there, right?
Well, I couldn't decode it. We were seeing activity there, but I couldn't use that activity to decode what it was that they were doing.
Okay.
Yeah. So yeah, we do see the hippocampus greater for space than for time, but we can't use that activity to decode what the space was that they were reproducing or estimating. So we know it's involved, but we haven't answered how it's involved. With respect to time, I can say that there are papers or studies that have lesion in the hippocampus and found that animals could still time, only if it was a time that they'd already learned. They couldn't acquire new intervals of time and learn them and go on to time them. So it does seem that the hippocampus is involved for kind of like a new acquisition, but not for, you know, kind of like things that have already been consolidated. But these are animals that could be overlearned, they could be overtrained, the things just could be entirely basal ganglia striatally linked at that point. So.
Forgive me if you addressed this earlier on, I came a little bit at the top, but it seems like one of the issues with time is that it's kind of confounded with strategy. So could you do manipulation where you give someone a dual task where they weren't able to? Did you talk about that? You're not-
No, I didn't.
I
No.
Would that be a possibility just to have them not sort of maintaining this counting or whatever strategy they're using to evaluate time?
Yeah, there's actually been a lot of work in this area. So the bane of any person doing a timing task is I don't want them to count. You know, I don't want them to count while they're doing this because there's been studies showing counting is not timing. You can use counting to help you do timing, but when you do, the behavior looks different. It violates the way, you know, we normally see timing happen, and it activates different parts of the brain as well when you're doing an active counting task versus a timing task. So how do you stop people from counting in your timing tasks? One way is you give them a secondary task to do, which is, okay, while you're timing, you have to do this articulatory suppression, or you have to also look for this target, or anything like that. The problem is, when you do that, you also then disrupt the timing task, and performance on the timing task kind of like gets all wonky and more variable, and they get bad at it. There was actually a study that came out, maybe like 2 years ago, that tested all the different strategies for getting a person to not count. And it turned out just saying don't count was about as good as like any of the, was about as good as you were going to get. Because they had like, you know, they were able to compare this to actively counting versus being told just don't count, to all the different other strategies like the dual task strategies, and just saying don't count seems to work the best. So now we all just cite that paper when we say we told people not to count. And that seems to work. I will actually, I will note that differences in strategy are an issue, and we have wondered if there is a way that we can tease apart what strategies are people using in these different tasks. But again, that's kind of difficult because same thing with the space and the distance task, people seem to jump from strategy to strategy, rather than kind of just picking one that they consistently do. Some do, but others just say, well, I tried looking at this, and others say, well, I tried looking at that. But that didn't work, and so I did this other thing. So it does seem to kind of jump around a lot.
So people know whether they're going to be reproducing distance or time before they start encoding.
Yeah.
Have you looked at cases where they don't know?
No, and we want to. So that's the next thing that we want to do actually, is have them do this task where just walk. But they don't know if they're going to have to reproduce the time or the distance. And so in that case, they got to hold both of these things in their head, but they don't know which one they're going to do. And so that is the next thing that we want to see, is like, well, how does that change this? And how does it possibly change like the brain mechanisms when you don't know what it is you're supposed to be reproducing? So that's exactly the next thing that we're going to start looking into hopefully soon. Cool. Yeah.
One last question. This is inspired by a poster I just saw at Economics where they were doing VR with black holes. So space and time are not linearly related. I wonder if there's anything that's some fun VR manipulation that you could use, given that it's all done in VR anyway.
Yeah, there was a study that came out last year. It was presented last year, it came out this year in Scientific Reports, that put people in a VR environment. They were playing like a kind of a shooting game kind of a thing, but it had a time manipulation where at random points while you're doing this thing, everything slowed down in slow motion, but you didn't. Like everything just like you're playing a shooting game and suddenly everybody's moving very slow, but you're fine. And then you can go ahead and hit all your targets, and then you're back again to like regular time. And they had them do this for something like 10 minutes or something like that in this weird warped time environment. I forget if it was under control of the subject or random. And they had them do a timing test before and afterwards. And they found that after doing this weird time warped VR, when people came back to reality, their sense of time was all messed up when they did this time reproduction. So it was the same task I showed you, except instead of reproducing a distance, they're just reproducing the length of a dot on a screen, They found that that got messed up afterwards, only after doing the VR task where time is all kind of wonky. So it does seem like you can do like a black hole kind of thing where we're going to warp and stretch time in weird sort of ways, and that will have a carryover effect into kind of like actual reality afterwards. So you can't, I think, you know, there's untapped potential here for doing cool and interesting things in virtual reality, and possibly also augmented reality as well with this. But we're just starting to kind of scratch the surface there. What did the black hole study show?
We have people going into and out of black holes and then do a pointing task to try to see if they could sort of retain direction within these black holes that were moving between levels of a game.
Yeah. Cool. Cool. Okay.
Very cool. Any good talk about space and time has to end with black holes. So that's
perfect.
That's a great spot to end on the sort of weird continuum of singularity that we've reached. So we thank Martin Weiner for his very and appreciate all your questions. Thank you for coming. Next Monday, our speaker will be Dwayne Watson from Vanderbilt University. This has to